Marburg and Ebola, the silent hell unleashed. The Germans opened the kidney of a monkey. They had no idea that, in that very moment, they were unleashing hell. In 1967, several hundred African green monkeys were shipped from Uganda to West Germany. In a quiet laboratory in Marburg, these animals were to be used for producing a polio vaccine that would save millions of children. As usual, the technicians donned their white coats and gloves. But suddenly, a knife sliced through flesh. Warm blood splashed onto rubber gloves. Within days, the technicians' senses began to collapse. A fever of 40 degrees Celsius burned through their bodies like fire. Agonizing pain seared behind their eyes. Vomiting and diarrhea were relentless. Fragments of intestinal tissue appeared in their excrement. The most horrifying stage had arrived. Pinpricks across the skin began to bleed. Blood poured from gums and eyes. Bruises bloomed under the skin, as if an invisible fist were hammering them from within. At the same time, in Frankfurt and Belgrade, the same nightmare unfolded. 3 outbreak sites, 31 infected, 7 dead. When scientists, trembling, dropped patient blood under an electron microscope, the laboratory fell into a deathly silence. The images revealed neither spheres nor rods. Instead, twisted, elongated snake-like filaments appeared. This was the Marburg virus. For the first time in human history, humanity faced a filovirus. If Marburg was merely a warning, then the outbreak 9 years later would be the filovirus' full-scale assault. September 1976, Yambuku village. Zaire was a medical desert, ravaged by extreme poverty. A church-run hospital had only 5 metal syringes. Every morning, the nuns used these 5 syringes to give hundreds of pregnant women vitamin injections. The virus hitched a ride on the needles, spreading silently and efficiently among the people. Weeks later, the village became a living hell. Patients burned with fever, vomiting, and suffering from diarrhea. Some began bleeding from their eyes and pores. 318 were infected, and 280 died. An 88% fatality rate. The survivors fled into the jungle, leaving the village nearly empty. Meanwhile, more than 1,000 kilometers away, at a cotton mill in Zaire, Sudan, another outbreak erupted. 284 infected, 151 dead, 2 massacres, happening simultaneously independent of each other. Different viral strains, yet from the same family. Local Congolese Dr. Jacquemain Yambuku ventured alone into the epidemic zone, risking his life to collect and send the first samples. Soon, a blood sample made its way to Belgium. Piot, a 27-year-old microbiologist, opened the package. Inside was a cracked thermos. Blood and ice swirled together. Under the electron microscope, he saw filaments almost identical to Marburg, yet distinct. He and his colleagues named it after a nearby river, Ebola. The Ebola outbreak was eventually contained using the most primitive methods, isolation and breaking the chains of transmission. It burned out on its own, only to flare again in sporadic outbreaks over the following decades. Scientists finally deciphered the virus's lethal strategy. Its first move was decapitation. Once inside the human body, the virus targets the immune system's sentinels with deadly precision. Macrophages, the body's guards, are turned into factories for the virus, which then sends out 2 powerful agents. VP35 blinds the cells, sensors keeping alarms from sounding. VP24 blocks the signaling pathways, preventing any reinforcements from arriving. Under this double lockdown, the body is fully invaded, yet the immune system remains unaware. And the final fatal blow comes from the body itself. When the immune system finally realizes it has lost, it does not retreat in order. Instead, it launches a suicidal all-out attack. Massive cytokines flood the bloodstream, tearing blood vessel walls. Clotting factors are depleted in an instant. Vessels become leaky sieves. The liver, kidneys, and adrenal glands suffer widespread necrosis. You are not killed by the virus itself. You die at the hands of your own runaway immune system. Death comes in as little as 7 days or as long as 2 weeks. For 46 years, humanity's response remained largely unchanged: isolate, trace, and wait for the fever to burn out. In 1995, during the Kikwit outbreak, Dr. Mo Yan proposed transfusing survivors, plasma, into dying patients. This attempt to find warmth in the ashes had little effect at the time, but it quietly planted the seeds of future breakthroughs. Yet the most crucial weapon, a vaccine, remained absent. No one wanted to fund full clinical trials. Ebola only killed the poor, with fewer than 3,000 infections total. There was no market, no profit. When a deadly disease occurs only in the world's poorest corners, the world chooses silence. In 2014, the silence was broken. Ebola emerged from the jungle and entered West African cities. 3 countries spiraled out of control, 28,000 infected, over 10,000 dead. And the virus even traveled by air to Europe and the US. The world's eyes finally turned. But at the moment the front lines needed help most, international aid was still on the way. At Kenema Government Hospital in Sierra Leone, Dr. Sheikh Humarr Khan was the country's only viral hemorrhagic fever expert. He dedicated his entire career to raising the only shield for the eastern provinces. He trained nurses, built testing systems, and fought hemorrhagic fever day after day in conditions of scarce resources. When Ebola overwhelmed Kenema, his hospital became the only refuge for patients. Isolation wards were full, protective supplies ran out, nurses fell one by one, and he knew the next could be him. Yet he continued donning protective gear daily, walking into the wards, until he himself developed a fever. On the 29th of July, without vaccine, medicine, or reinforcements, Dr. Khan fell on the darkest road before dawn. What Dr. Khan never received, others later developed. That autumn, Dr. Chen Wei of the Chinese Academy of Military Medical Sciences led a team to Freetown, where the city's medical system had collapsed. Flights were grounded, fear hung in the air. Under the blazing sun, Chen Wei's team built China's first high-level mobile laboratory. Diagnosis time shrank from days to hours, bringing more than just testing capability. China's adenovirus vector vaccine L5 EBOV completed clinical trials in the epidemic zone and was approved in 2017. 3 years later, the same technology was directly applied to the COVID-19 vaccine Convicia, from project approval to clinical trial in under 2 months. Without the magic sword in Freetown, China's later speed would not exist. Canada's Cobinger team developed the rVSV-ZEBOV vaccine, achieving 0 infections 10 days post. Vaccination in Guinea, approved in 2019. It became humanity's first Ebola vaccine, and it took 25 years to honor Dr. Mo Yan's 1995 rough intuition, saving lives with survivors' plasma, which ultimately led to monoclonal antibody therapies. A Kikwit survivor's memory, the immune memory of a single B cell, traveled a quarter century to become a weapon saving millions. In 2020, the FDA approved treatment that turned a 1976 88% fatality rate into over 90% survival for early patients treated with antibodies. It took 44 years to reverse those numbers. Deep in the jungle, the virus still clings to a fruit bat's cells, quietly waiting for the next chance. But humanity waits too, armed with vaccines and antibodies. The fruit of half a century of sacrifice. Humankind has never truly won, but has never surrendered.